Good morning, good afternoon, everyone. I think we can probably get started. Everyone can see my screen, right? So in terms of agenda, I'll just go through, an executive summary as I usually do, and then we'll go into, discussion about the data, some recent data on 2281. We'll talk about the hepatocyte study that was done with 2281, and then we'll go in to the efficacy study. So, we haven't had a meeting now for a few months because we've really been kind of focused on these two mouse models in order to hopefully make a decision on which compound to move forward with. So we'll be discussing some of those that data in a lot of detail. And then move into, what are the next steps in picking a compound to move forward with. So, we do have some, we basically caught up with all of the data on the three development candidates, right up to what we had historically with 2009. So for 2281, I would say you'll see the data, but there was really no red flags that came out of that in vitro data that we accumulated for 2281. We did decide to do a metabolic stability and MET-ID study with 2281, with the thought that would be the development candidate. So I will go through some of that data for that study, and then really we'll finish off with these two in vivo efficacy studies. So to give you an update on the results from Maria Soli's lab, some of you have already seen this data, but with 2009, it basically recapitulated that study that was done pre-COVID. However, with 2281, we didn't really see any extended survival with 2281 in this model. The RCAS model, which Rebecca will be describing, we're basically still in the middle of that study, so she'll give an update on sort of where we are in terms of how many mice have been dosed and how many are still looking to be recruited in that study. So this is the usual table. Not a lot of new data, but I will say that we did run a kinase selectivity panel, the Eurofins panel with 2281, and as you can see, it's quite selective, on par with 2009 and 2308. CYP, no concerns with CYP inhibition with 2281, was negative in the AMES test, but again, this potential red flag for this compound where we didn't see any extended survival with in this patient-derived xenograft model. However, with 2009, we do see that modest extension of survival that we saw previously. So this is the metabolic study, or results from the metabolic study that we ran. So this was a cell-based assay run in hepatocytes in 6 different species. And really what you can see is, at least in dogs and certainly in mini pigs, we do see rapid clearance relative to humans or mouse or even rats. So basically in one of these other non-human, non-rodent species, we do see a little bit higher clearance than what we see in the rodent species. So what I've done here is basically gone through the metabolite ID that was run, and anything that showed up over more or less 1%, I've kind of flagged here in green. Really the only one on this slide that I think has any that we should note is this M7. It does only show up, at least that we see in monkeys, but it is a potential red flag as it is an acetylglucosamine conjugate. So, if we do want to look at this further, we can certainly explore this further in some in vivo studies. On the second slide, not surprising, M14, we do see that show up. That is the demethylation to 2308, and we see that basically across the board. But I do want to highlight M17 and M18 as a potential red flag. So what we see is oxidation on this piperazine ring, followed by what appears to be demethylation to this N-hydroxyl piperazine adduct. So again, if we want to kind of explore this further, there's a few options. We could certainly run 2308 in this study and see if this hydroxylamine shows up. If we want to actually make it synthetically and run it, this should be fairly straightforward to synthesize. And secondly, we probably should run a CYP induction on the compound that we're going to move forward with, either one of these three. But certainly we can look more in vivo if any of these are something that we need to be concerned about before choosing a tox species. Does anyone have any questions on these on this MET-ID study?
Peter, hi, it's Mike Briggs.
Yes, Mike.
Do we have CYP-TDI data on any of these compounds?
No.
So I understand any risk around potential reactive metabolism. So that would be something to look at, I think.
Yeah, for sure.
Yep.
Okay. Running through this pretty quickly. Rebecca, maybe I'll turn this over to you.
Hiya, yeah, sure. So for those who haven't seen this data, this is the in vivo efficacy in HSJD-DIPG-007, which is a patient-derived model, which was run in NSGs by Maria and Amina in the Ziegler lab in Australia. So they had a vehicle and then ran M4K2281 and 2009. And as Peter's already said, we saw that moderate but significant shift of in survival with 2009 of about 10%, but we didn't see the same thing for 2281. So what aren't on these slides and what was shown at the other meeting was that we have got PK back for these, and there wasn't anything suspicious in NSGs that came up to suggest one's getting in and one isn't, because these are in two different vehicles. So I think 2281's in sodium citrate and the 2009 is in the methyl cellulose. So there's nothing that we can see from the PK that we ran before this study was conducted. Maria still has all the brains from these mice, and so if it's something that we want to look at specifically in these treatment arms, then that's an option. They also did luminescence readings, and so in the middle, kind of bottom middle panel is the data across all the different time points that they tested, and then on the far right is the week 3 snapshot, which shows that you do get a reduction with the M4K 2009, but not so much with 2281. So this was, the cells were injected, 4 weeks later they started being dosed, and they were dosed for around 4 to 6 weeks, which is slightly different to the setup that we've got in the lab for the RCAS model, which I think might be on the next slide. Yeah,
so they do have, they did keep brain tissue samples. Yep. So that's something that they could run in Australia, or would we want them to?
Yeah, I mean, I'd have to double check whether they'd be able, they've got the capability of running PK, or, just the time to do it. We could always get them shipped to us and we could have our PK team have a look at them. But they're stored and frozen, so the options there, I'm not sure they're running them themselves. I'd have to double check.
Okay.
Yeah. Sorry, I may have missed this. Did you say the two compounds are in 2 different formulations?
They're in two different vehicles. Yeah, 2281 wouldn't go into the methylcellulose. It kind of precipitated out.
Okay, and what is the vehicle then?
Sodium citrate. So we have done all the tolerability that we tested before these efficacy, and the PK was done in those two separate vehicles.
Okay, thank you.
Yeah, I think I have those slides, I think, in backup if you'd like to see them.
And then the 2308, we've also run. So on the next slide, you'll see that that's also in methylcellulose. Next slide, please, Peter.
Okay.
And so this is where we're at with the ARCAS model. So this is Oren Becker's ACVR1 mutant mouse model. So this study is set up slightly different, and I think I've mentioned this before, but just to recap that what we're trying to do with this experiment is we inject the mice and then we wait for them to reach a threshold of BLI signal before they get enrolled into dosing. So we've noticed that the cells can all be injected, and some the BLI kicks up after 2 or 3 weeks, and some it's 5 to 6 weeks, and so then that kind of affects the survival. This actually it seems to be working quite well. I think it's quite a bit more work, and I think Deanna's on the call, so she's been the one doing all this, so she should get all the credit for this work. But we run in all three, so 2308, 2281 and 2009. These are in now the black sixes because this is a immunocompetent model. And so on the bottom, we're just trying to show you the BLI signals for all the mice in the different treatment arms and kind of where we're at. So because we're injecting these in batches, some of these mice are 50 days post-treatment, and some of them are 30 days post-treatment. So we're injecting once a month. So I'm just trying to, hopefully that's coming across here, that even though there's currently six mice or 6 or 7 mice per group, they're not all at the same post-dosing stage. Currently, we're aiming for 12 mice per group, and Deanna has injected a whole bunch of extra mice today. So we are getting there. Oh, sorry, Peter, I sent you, sorry, it was a bit late. I sent you another slide. The X's are actually not quite right here. If you go on to the next, oh no. We've lost 3 vehicle, 1 M4K2308, 2 of the 2281s and 1 of the 2009. So these X's were meant to indicate dead mice, but basically the ones that have all got the higher BLI are the ones that have been culled. This is actually the reverse X's. But it looks like when we look at when we plot the BLI for all of the mice, it looks like 2308 and 2209 are actually doing better at keeping that BLI down, whereas 2281, that BLI is coming back up, which is a bit similar to what Maria was seeing. I think on the next slide, there's just a table of the summary, but again, that's not quite right. I can resend the slides and share. So I think all the ones that say yes in 2281 and 2009, it's actually the reverse. So the yeses are no's and the no's are yeses, sorry.
Okay.
Yeah, sorry. So they're better though, I think.
So these are still, all these animals are still alive.
Yeah. So okay. It's there's only one has died in 2308, the top 2 have died in 2281, and it's only that second mouse that's died in 2009. We haven't been plotting survival for this just yet until we've got the full complement of mice that we're hoping for per treatment arm. But it looks like, at least in terms of the BLI, that 2009 and 2308 are doing something compared to vehicle.
Okay.
Yep.
So that's basically in.
Yeah, I mean, if it helps, it's a bit confusing for everyone. I've got the slides here. I could share them if that helps, but it's the kind of the opposite of the X's here.
Okay. I mean, maybe at least for myself and maybe for others, can you describe what we're seeing here with the different on the color scale?
For us, the signals reach the threshold. We've got kind of 2 times 2.5 times 10 to the 6, and that's based on historical data where we've tested these and see kind of where the BLI kind of kicks up, and then about 4 weeks after it reaches that threshold, the mice start to die, and we are seeing that quite reproducibly for the vehicle mice. So in these images, blue is signal, and you can see for some of the mice, so mainly if you look in the treated group, so the first two for the M4K2308, you get signal to start with. So the pre-treatment scan before anything starts, before they get enrolled, you can see the blue signal. And then with treatment, you can see that this signal goes down. So you can kind of see they lose that blue signal. The ones where the mice have been culled, so that third one in for 2308, you can see the signal goes up a bit because it kind of gets more greeny in the middle. So as you go up to green and red, the signal's getting higher, and that mouse was culled. For 2281, I think it's the first and the third column, those mice have been culled, and you can see that signal goes up, whereas for that one that's currently got the X underneath it, it goes down, and that mouse is still alive. So it's just we're trying to track this signal over time so that we can get two readouts. We want to know what the survival is, but we also want to know kind of the growth of the tumor before you just end up with dead mouse.
Okay. And so you do this scan like about every 2 weeks or so? Is that?
Yeah, so initially for enrollment, it's we do it at day 7 and 10, and then they get enrolled on day 14 because this model's fairly quick compared to our patient-derived models. What's tricky, and I think Deanna's showing here the images just for consistency, but I think the mice are getting imaged once a week, but because they're injected on different days, they're enrolled on different days, I think Deanna's put the images here that kind of match up for the whole set, but I'm pretty sure they're getting scanned once a week. Okay. So for each individual mice, we'll be able to plot that over time, but just because of how the experiment's set up, that's what we're showing here. And
then based on that scan, like this mouse would be just, it's called based on the scan? Or
No. So actually we have a whole set of kind of neurological symptoms that would trigger the mouse to be culled. Okay. So this BLI is just kind of because we've labeled the cells and it's extra information for us, and we might be able to get some stats on it, because it could be that initially the compounds do have an effect and then they come back up, and maybe that's why you're seeing the delay. So it's just nice to be able to capture the dynamics of the tumor growth, but it's not they reach a certain BLI and then we cull, but generally it's pretty consistent. So when they're starting to show neurological symptoms, it tends to be the similar BLI value.
Okay, so when you're calling them, it's not just based on a particular day just because of that scan. There's other things that are going into it. And then
there's a whole raft of neurological symptoms that Deanna and the team look out for.
Okay, I see. Thank you.
Are you taking any PK samples from this experiment at all?
So no, we haven't. Well, we take all the brains, but we weren't planning on using them for PK. We've done all our PK up front for the single agents in the 2 different strains, and it all looked fine. I don't know if you think that we'd need PK for this. It's not typically something we would do if we've already run the PK study up front, unless maybe something really weird was happening, but it seems to be on track with similar results to what Maria had seen, so it's not overly surprising.
I guess I'm just interested in the differences between the compounds, because I suppose we're probably quite surprised that 2281 isn't working as well as we'd expect. So it might be interesting. Yeah. I realise we can't do it for every mouse and all the time, but it might be interesting to look at a couple of samples to see whether there's something going on there about exposure that we're not expecting. I don't know.
Yeah, because you're right in that the PK that we've run before, they are on non-tumor bearing mice. So
can I just say something? It's Deanna.
Oh, hey.
Hello. Just to say that for some of the mice, we are also planning to do some MALDI-TOF. So on the tumor-bearing mice, we're going to look for the drugs. We're still not sure they're able to detect the drugs. We're still at that stage, but I'm going to collect some brains, and we're going to see at the same time point that we've done for the rest of the PK study, so 2 hours after dosing.
So that's kind of like sections, isn't it, Dan? You take a section of the brain with the tumour, and they should be able to see where the drug is.
Yeah, and if it's in the tumor, in the brain, around, and we should be able to at least have an idea if there is a difference between the drugs or not. I think that would be helpful.
Sorry, Rebecca, going back to Charlotte's comment, if I would say if you ever have to cull a mouse, that may be an opportunity to take a terminal sample, and it doesn't matter at what time you take that terminal blood or brain sample, or both if you could, just make a note of what that time was after the last dose, because I think what these types of studies, just as a sort of for a project team, we've already heard about the metabolism, and we've got some potential flags in potential metabolic pathways. What we've got here on repeat dosing is basically like a tolerability study as well as a pharmacology study ongoing. So we may want to just sort of monitor levels of circulating compound that may indicate whether there's, Peter made a comment earlier about we may need to look at some CYP induction endpoints. Well, we would see a flag if there was a change in the pharmacokinetics over time. So, you know, I wouldn't say change your protocol now to start including samples, but if you ever have to cull an animal, I think it would be useful to get that information. And like I said, time point is irrelevant as long as you make a note of time after last dose within an hour or two. You know, it was 2 hours, 4 hours after the 30th dose. And you know, the PK people can then go back to your single dose PK and look at the level that's measured and say, oh, that's very similar to what we might have expected. Nothing has changed remarkably over repeat dosing. So it's just an opportunity to get a feel for what may happen on, we call it time dependent effects on pharmacokinetics, i.e. repeat dosing. So it's just a like polite request, really.
Yeah,
if there is a chance, even if it's just 1 or 2 animals, it's just another piece of information to help.
I mean, we do collect. We've collected, but I don't think we've collected blood as part of like when the animals are culled for neurological symptoms for survival. But the brains are definitely collected, and we do have certain assays that we use those brains for, a bit like the Malditoff, like Deanna's saying, and PK markers, PD markers, sorry. But if there's, you know, I'm sure we can put a couple of those brains that we've collected for this, and like say, just take a note at the time. I'm just trying to think, Deanna, with the PK study, is that at the end of the tolerability or is that separate? Because the tolerability is run for 3.5, 4 weeks, right?
Yeah, it's at the end of the tolerability after the 6 days of treatment.
So they get quite a bit of treatment, and then it's already 2 hours post last dose. So it's not like the mice have only had two hours of drug.
What you said about the brains is interesting then, because it sounds like you're going to have more brains than plasma samples. We have an idea of how these compounds distribute into the brain. So we could use brain level as a surrogate of what's happening in plasma. For example, if the brain levels are as we might have expected, we would assume underneath that the plasma's not changing too much upon repeat dosing. But I think it's just, you know, if we can get that information and just have a look at it from a PK perspective, it just informs us whether or not there's anything changing. What you're doing is you're trying to see if there's any potential issues when you go into safety studies with repeat dosing and changes in pharmacokinetics and things like that. Okay.
I guess with this one though, it would be, you'd be banking on the fact that this drug is still in the brain 2-3 weeks after the last dose, because that's kind of when they're getting cold.
Oh, is that? Right? I'm thinking. The culling.
Not quite. So for these ones, so for these, they're all still under treatment. That slightly longer models, and maybe data that we've shown before with the patient-derived models, we are restricted. We can only treat for 20 or 24 weeks. And so if the model's very long and we start perhaps a little bit too early, then there is an amount of time where those mice will be not treated and then still culled for survival several weeks later. But for these, they're all still on treatment. So they're all dying under treatment. Yeah.
Okay, thanks. I didn't understand that part. Okay.
Yeah. So for these, maybe I should adjust this actually.
What Mike is saying.
Yeah. Yeah. And we can, you know, maybe send an email of what brains we have and what we've got available to look for what you're asking for. So.
Yeah, and I just before I forget, with Diana's comment about the brain slices, for at least for 2281, it would be important to look for 2308 in that particular study as well.
Yep.
But
it's not something that we've done before, Deanna, right?
No, we haven't done, and we still have to send the drugs to see if they actually can see it, to spot them and see in the MALDI. But if that is okay, then we'll go ahead and collect the brains for this.
Okay.
But for the mice that are currently under treatment, I can definitely collect the blood. That's not a big problem. But all the brains that I've collected so far have been fixed in formalin, so they wouldn't be suitable for the PK. So we'll just have to rethink of how we maybe we cut in half or something, and we do one thing for PK and another one for formalin to look at the tumor burden. But yeah, for the mice that are gonna be collected, later on, I can definitely collect the blood.
Okay. Any other comments for Rebecca and Diana? Well, thank you so much for this update and for clarifying all my questions.
Yeah, and we'll keep you posted as it comes through.
Okay. So lastly, it showed this was what we, oh, that's a typo there. Basically at our last meeting where we were in the timeline, and we've had to obviously adjust this timeline slightly because we're still not at the point where we've chosen a development candidate. So it really hasn't impacted a lot of these downstream activities as yet, but obviously we're getting close. The longer this is delayed, then this will start impacting some of these later stage activities. But really, I think for next steps, it's, can we choose a development candidate maybe in the next 4 weeks, or do we wait a little bit longer for this RCAS study? It's looking like, at least in now 2 efficacy models, that was I think a favorite of mine and other people on the team, 2281 is starting to maybe fall out of favor based on those studies. But I think maybe we just wait a little bit more to see how that performs in that study, unless other people have a different opinion. But once we actually pick that development candidate, then I think we need to start process optimization. So the chemistry side, that's really going to need to ramp up quickly in order to have enough compound to really do a lot of these more compound intensive ADME and DMPK studies that we'll need to do. But once we actually do those studies, next thing is to engage some toxicology and consultants to kind of help us start planning for a toxicology study. That's essentially what I've had on the docket for today, but certainly interested in hearing other people's comments on where we are with the development candidate. Certainly, I think we have enough information now that certainly 2009, we have enough data to have that as a development candidate. I think the question is, do we pull the trigger on that within the next, even today, or do we take a look at 2308 and 2000 or 2281, continuing on in this RCAS study? I think for me, I think I'd like to see some more data from Rebecca's study before I make before we make a final decision on the candidate.
Peter, it's Mike again.
Yes.
What about getting some cross-species MET-ID on 2009? I don't think we have that. So we've seen some flags with 2281. Given the metabolic pathway of 2281, we would expect some of those flags to be carried forwards into 2308.
Mm-hmm.
So it's there, my mind is, if you want to start talking to toxicologists, and if we go down the route of 2009, they'll ask questions about metabolic profiles in different species, which species, you know, would we select, and we can have a look at these, you know, what the pathways are, start looking for any evidence of, we call them smoking guns, reactive metabolites or not, or glutathione adducts and things like that. So I think I would, you know, throw that into the hat from my perspective for the team.
I think that,
think about, and the TDI maybe on all three, because it is a quick study. TDI.
Even the Med ID study was a pretty fast turnaround with the group that we went with. So
but the TDI gives you that some feel whether or not you've got any potential for reactive metabolites. If you see a change in the IC50 over with pre-incubation, and if you saw some flags in the metabolic pathway study, you then start to say, okay, there's a bit of a risk starting to show here. So I think that certainly the TDI for all three of them, if we're happy to do that, and then the Met ID on 2009 would be my, if we could get any more data within the next 4 weeks type of thing to help with that discussion while the pharmacology studies are still ongoing.
Okay. I think that's a really good suggestion.
Given resource constraints, I mean, can we make sure that we're not preferring 2308 first before we invest in that? Because we already have enough data on 2308, I think, from the 2281 Med ID study, don't we? I mean, they've been metabolized pretty comparably. So if there's some reason to prefer 2308 anyways, let's hold off until we know that. We're I think we're still short. We're still short on cash to do everything we need to do, right? So
I think Mike's point is we would expect to see some similar metabolites if we run 2308. So it's something, if we choose it, we'd have to do it, but I don't think we need to do it now. But 2009, I think it would be good to run that so that we have a head comparison of 2009 versus at least 2281 and potentially 2308 in a metabolite profile.
Yeah, but I'm saying we're going to get efficacy data here shortly. If that suggests that 2308 is superior for some reason, then we wouldn't bother with 2009, would we?
Well, I think to Mike's point, it kind of mitigates the risk if we do see some red flag metabolites, which, you know, I wouldn't say they're huge red flags that we've seen, but we still would need to do some additional studies. And I think TDI is certainly a great suggestion on from what we've seen from the Med ID on 2281 and extrapolating that into 2308. And if we run,
I'm just reticent to spend another $20,000 before we even have a clinical candidate, because we're going to run out of cash. We don't have enough money to do the next phase. So if there's any way we cannot do that until we absolutely have to, I think we got to look at that.
Okay. Yeah, I mean, we can certainly take that offline and discuss it in more detail. So otherwise, I don't have anything else right now. I think we'll certainly look to Rebecca's group to continue to feed us more data from the study as it's ongoing. And then discuss the merits of doing some additional ADME work prior to the next meeting, if that makes the most sense, both scientifically and I guess fiscally. Yeah, so if there's no further comments, I wish everyone a good day, and we'll talk to you soon.